हाँ जी सर, स्ट्रीमिंग ना बंद हो रही है, आज मैंने फिर से स्टार्ट कर दिया है। हाँ हो रही है सर, वो तो हो रही है। हाँ हाँ, कल से कर देंगे। हाँ जी सर। जी जी सर। ओके सर। Hello, hello, welcome back, welcome back. Sorry, I had network issues today, but still, I was thinking that it will not happen today, the class, but still, we are able to manage it and we are doing it in a very healthy manner. So don't worry, we will finish it soon in a good way. So just be with me. So I will start with previous from protein itself again, so that you have missed it before. It's So students, please join in. Then we can start again. So I'm really apologize from the bottom of my heart for providing such a lack of disturbances today. But from tomorrow onwards, everything will be fine as smooth. This is my guarantee. It's just for today because of I'm living in the outskirts of my Delhi. So, but still, I was able to get internet, and I want like today was the first day, so I don't want to miss you all. So I tried my best that I could teach you uh, the very basics of molecular biology. So please join with the new streamings. I hope new streaming is working fine now. All is good. Yes, yeah, there are platforms like Zoom and Google Meet, but there are around 400 students, and it's not possible to, but because in Zoom and Google it's only 100 are allowed. So because of 400, we have to adopt this method. So that's why. So just it's just for today. This we are having this problem. We will be fine with tomorrow with that. No problem will be there then tomorrow. Thank you. Thank you so much for understanding. I'm trying my best to be louder. It's something. Maybe the room is so big, so my voice is not able to get you. But I'm loud. I'm speaking so loud that the voice is coming after striking the wall. Actually, so so let's see. So let's focus on learning rather than this small networking problem. They will not stop us to learn more. So that's what I'm here for, to teach you the best for the best things. Yeah, it's kind of breaks. That's good. That's improvisation. Yeah, I'm using earphones. I'm using earphones. Uh, to make it. It's just the room is big, so my voice is not. Where I'm teaching in my room, it's a solid like soundproof room, and it's very and there's no network issues there. Also, it's just for today. I want to make things done good way. So, but I did somehow. I have to say. But let's finish it with a good end. So how many students do we have so far? So let more people join us. Uh, okay, that's good to hear that I'm audible. That's awesome. So I will not waste much time before it goes off. Let's start with another streaming. So proteins are made up of amino acids arranged in a linear chain, folded into globular form. The sequence of amino acids in a protein is defined by the sequence of gene which is encoded in the genetic code. And there are around 28 standard amino acids. So here you can see a polypeptide chain of amino acids, all bonded together in the post-translation modification with the quaternary structure having beta-plated sheet, alpha sheets, manganese ions in center, heme group is also there. So all are being together, and these proteins could be sourced, could be available from the groundnut, or you can get it from the red beans, uh, fish, eggs, meat, chicken, pulses, white chickpeas. So all these are the various sources of uh, things. So basic players in molecular biology, DNA, RNA, and proteins. What they do is the DNA replicate itself. It's called as replication. Then DNA makes RNA, RNA makes protein. Where DNA is making RNA is known as transcription, and RNA is making protein is known as translation. And this is known as central dogma, central to the life. So DNA replication is a basis for biological inheritance, is a fundamental process involved in all living organisms to copy their DNA. In the process of replication, of DNA each stranded of a DNA molecule, they serve as a template for reproduction of complementary stranded. Two identical DNA molecules have been produced from the single double-stranded DNA molecules. So I have to keep check in between if everything is okay. Okay, good. So this is DNA polymerase. So here you can see a single strand of DNA which has been held together, made it into unpaired chain by topoisomerase, and helicase is the one which is breaking up these polymers into two parts and creating two strands, leading strand and lagging strand. Leading strand is the one which is automatically being produced with the help of DNA polymerase and producing a very nice polymer strand with a 5' to 3' end. Whereas the lagging strand is the one which having some okazaki fragments which are being joined together with the help of DNA ligase and also help with the RNA primer and DNA primers. So in a cell, DNA replication begins at a specific location in the genome called origins. Unwinding is done and leading to the replication fork. So it's a replication fork you can see like this. In addition to DNA polymerase, the enzymes that synthesize that new DNA by adding nucleotides match to the template strand, and a number of other proteins are associated with the fork assist in the initiation and continuation of DNA synthesis. Cellular proofreading that ensure near perfect fidelity of DNA replication. Transcription is the process of creating an equivalent RNA copy of sequence of DNA. Transcription is the first step leading to the gene expression. DNA is producing transcription is producing RNA and RNA is producing with the reverse transcription is a DNA. So DNA transcription and DNA sequence is read by the RNA polymerase which produces a complementary entry parallel RNA strands. So we already know the complement in this instead of uracil we have thymine in this case. So transcription process, the stretch. So transcription process, stretch of DNA transcribed into RNA molecule is a transcription unit and it codes at least one gene. If the GNA transcribed encodes for a protein, the result of transcription is messenger RNA, and this messenger RNA will be used to create that protein by the process of translation. Alternatively, the transcribed gene may be encoded for either RNA or tRNA, other components of the protein assembly process, or other ribosomes. So DNA transcription unit encoded for protein and regulatory sequence that direct the regulate synthesis of that protein. So DNA is read from 3' to 5' and during a transcription, the complementary of RNA is created from the 5' to 3' direction. Only one of the two DNA strands called the template strand is called transcription because RNA is only single strand. The other DNA strand is called the coding strand in that case. So here you can see that there's a promoter sequence A, C, T, G, T, T, A, T, T, A, T, T, A. There's a start signal from there and your RNA polymerase has been attached there. So there's a complementary DNA started to produce on the site. So like this self-complementary sequence. So RNA polymerase is removed and hence at the end you have a mRNA out of your DNA. So this is RNA synthesis and processing. So there's exons and introns. So introns are the one which are not coding for mRNA and they are like removed during the RNA splicing and then you have mainly exons are left, exon 1, 2 and 3. So reverse transcription viruses replicate their genomes by reverse transcribing the DNA copies from their RNA. These DNA copies are then transcribed to new RNA. Retrotransposons also separate by copy DNA and RNA from one another. So reverse transcription, HIV RNA, cytoplasm, HIV DNA, so they're all process of reverse transcription. So in general, we can see that the DNA is producing RNA, RNA is producing protein, and in the reverse is special case where RNA is producing self-replicating with a reverse transcriptase and also producing DNA in that case. So translation is the first stage of protein biosynthesis. In translation, mRNA produced by transcription is decoded by the ribosomes to produce specific amino acids or polypeptides that will later fold into the active proteins. Translation occurs in the cell cytoplasms where the large and small subunit of ribosomes are located and bind to the mRNA. So here you can see that we have seen in the video itself also how translation process was going on. So you have your ribosomal RNA, your valine has been attached there before, tRNA is removed, then valine, then alanine comes, then glycine will join it, and hence your proteins of amino acids will keep producing like this. So this is the direction of ribosome advance in that direction, and codons for these amino acids are attached here, and this is your ribosome, and below is your messenger RNA, and their various transfer RNAs are coming like this. So entire ribosome will bind to the outer membrane of rough endoplasmic reticulum and release a nascent protein polypeptide chain inside for later vesicle transport and secretion outside of the cell. So genetic code. So this is the genetic code that you can make by writing UCAG, UCAG on this side and UCAG, 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 UCAG. So UUU phenylalanine. So you write first alphabet from here, first alphabet from here, first alphabet from here, U. So you join them together, UUU, and it's become phenylalanine. And then UUC, UUC, it's become also phenylalanine. So write like this, you can write all the amino acids group and group them together and their name will be like this. Like GCU will be alanine. So methionine, AUG is your methionine is a start codon. And your stop codons are UAA, UAG, UUGA. These are your three stop codons. So what is genome? Genome is an entirety of organisms' hereditary information. Is encoded either in the DNA or for many types of viruses in RNA. Genome includes both genes and non-coding sequence of the DNA. So comparative genome size of organisms. So Homo sapiens. So this is the comparison has been shown with the various chromosome numbers from Homo sapiens to the bacteria with the size base pair numbers and the chromosome numbers in them. Why genome analysis? The prediction of, let me keep checking you students if everything is fine. Yeah, all is good. Why genome analysis? The prediction of genes in uncharacterized genomic sequences to obtain the complete sequence of as many as genomes as possible. For genetic modification, for modification to develop new varieties at a 5-star rates like BT cotton, BT brinjal. So on this note, let's take a 5 minutes break, or we continue because we never know how when the internet will go off. We will have a problem. We continue till the end. We might finish earlier then because of that. So tools used in molecular biology. So the basic principle is that DNA, RNA, and proteins can be separated by means of electric field. Your DNA and RNA and protein could be separated by the electric field in agarose gel. Electrophoresis DNA can be separated on the basis of size and by running the DNA through the agarose gel. So here you can see there's a negative electrode, positive electrode, DNA binds separated by the size. So DNA bands are separated because of the size. And DNA is a negative charge because of the phosphate bond in it, and they move from the negative side to the positive side. So DNA bonds are exposed to the film, and we can check them under UV light how they are visible. So in agarose gel electrophoresis, DNA can be separated on the basis of size by running through the agarose gel. Protein can be separated on the basis of the SGS page gel or on the basis of the electric charge by using what is known as 2D gel electrophoresis. So polymerase chain is an extremely versatile technique for copying DNA. PCR allows a single DNA sequence to be copied or altered in the predetermined way. PCR has many variations like reverse transcription, RT-PCR, qPCR, quantitative PCR, which allow quantitative measurement of DNA or RNA molecules. So PCR analysis looks like this. You have template DNA, you have first cycle, then you have second cycle, you have third cycle, then you have fourth cycle. So they are in the PCR, your copies are increasing each time. So 2 to power 2, 2 to power 3, 16, 32, and exponential amplification is done in this way. The process follows the principle of DNA replication. So primer is a strand for nucleic acid that serve as a starting point for DNA synthesis. These primers are usually short, chemically synthesized oligonucleotides with a length of about 20 bases, and they are hybridized to target DNA, which is then copied by the polymerase. So minimum primary length used in the most application is 18 nucleotides, starts at the 3' end of the primer and copies at the opposite strand. In most cases, natural DNA replication primer for DNA synthesis and replication is a strong strand of DNA. So what are the applications? It could be assessed in DNA sequencing, phylogenetic analysis, parental testing, genetic mapping. So all of these are various applications of PCR. So let's continue. So macromolecules blotting and probing. So this is what you can see, a genomic DNA, and we cut it with the help of restriction endonucleases and then put them on the agrogel electrophoresis with the high molecular weight to low molecular weight from negative to positive electrode. Then the blotting buffer has been created. Agarose gel is there. Nitrocellulose membrane or nylon membrane are put up. Then they are went for the nitrocellulose membrane. They've been transferred from gel to your blotting member. Your gels, your bands are being transferred to the nylon membrane, and they are probed with the 32P radioactive phosphorus. Then they're exposed for the X-ray. So this way you can do the blotting. Then there is a southern blotting, which is done to check the DNA sequences, separated by gel electrophoresis and then transferred to membrane by blotting by capillary action. The membrane is then exposed to a labeled DNA probe that has a complementary base sequence to the sequence on the DNA of interest. Less commonly used due to the capacity of other techniques such as PCR. Southern blotting are still used for some applications such as measuring transgene copy number in transgenic mice or gene knockout embryonic stem cell lines. Then there is a northern blotting. Northern blotting is to study the expression of proteins or specific type of RNA molecules as a relative comparisons among the set of different samples of RNA. So it's mainly done for the RNA, and southern blotting was done for the DNA. That's the main difference. And western blotting is done for the protein case, which is also known as SGS page or sodium rhodosyl sulfate polyacrylamide gel electrophoresis. in which gels are then transferred to nitrocellulose membranes, nylons, or sport membrane after the transferring between the two plates of sandwich. And this probe with a solution of antibodies. Antibodies specifically bind to the protein of interest, and we can check them with the help of chemiluminescence or autoradiography. Using Western blotting technique allows not only detection but also quantitative analysis also. So these were something about the blotting, southern blotting, northern blotting, western blotting. Then there's molecular markers based on the naturally occurring polymorphism in DNA sequence. So genetic markers are sequence of DNA which have been traced to specific locations on the chromosome associated with the particular traits. It can be described as a uh, variation that can be observed. So they're also called as mini satellites. So here you can see genetic markers as RFLP, AFLP, RAPD, VNTRs, SNP, STRs, SFP, DART, RAD markers. So all are these various genetic markers, their full forms. So there we will deal all of whatever the techniques that I'm showing you here. We will deal each of them in detail. So this is an introductory class. So where first we have discussed about DNA, RNA, and the various molecular biology basic concepts from transcription, translation, central dogma, reverse transcriptase, PCR. So all these techniques after the basics we will do all these techniques also in details in future. So this is just giving you a short hint or short things how things are happening. So five conditions that characterize a suitable molecular marker must be polymorphic, codominant, inheritance, randomly frequently distributed throughout the genome, easy and cheap to detect, and reproducible. So several different applications are genetic diagnostic kit, study of genome, paternity test, measure the genomic response to selection in the livestock. Then take the RFLP example. It involves a fragmentation of sample of restriction enzymes, which can be recognized and cut DNA wherever specific short sequence occurs. RFLP occurs when the length of detected fragment varies between individual and can be used in genetic analysis. So it has variant and codominant. Main disadvantages requires a relatively large amount of DNA. AFLP is this the one which is used to amplify restricted fragments, length polymorphism, which analyze your material of around 1000 folds, and it can be used for comparisons between closely related species only. Advantages are relatively inexpensive, high variable. Disadvantages are markers are dominant. Presence of a band could mean the individual is either homozygous or heterozygous recipients. RAPD, random amplification of polymorphic DNA, is a type of PCR actually, has a segment of DNA that are amplified at random. Advantages are they are fast, relatively inexpensive. Disadvantages are markers are dominant. Presence of a band could mean the individual is either homozygous or heterozygous. Data analysis are more complicated. Microsatellite polymorphism, SSR, or simple sequence repeats. Microsatellite simple sequence repeats or short tandem repeats are repeating sequence of 1 to 6 base pairs of DNA. Advantages are highly variable, fast evolving, codominant, relatively expensive, and time consuming to develop. A single nucleotide polymorphism is a DNA sequence variation occurring when a single nucleotide ATCG in the genome differs between of species chromosomes in an individual. Used in biomedical research, crop and livestock breeding programs. STR, short tandem repeats, is a DNA sequence where a pattern of two or more nucleotides are repeated, and repeated sequences are directly adjacent to each other. So they are in length of 2 to 16 base pairs, used in forensic cases, used for genetic fingerprinting of individuals. Now comes the last part, how the principle of DNA and isolation of purification is done. Uh, okay, Bache, all these techniques that we have studied today, we have done basically all the techniques of northern, southern, western, some of the RFLP, AFLP, RAPD markers. So we will deal in details soon also. So that was just to give you just hint how these things are done, just to have an overview of it. But we will deal with them soon in a more enhanced manner in each lecture. So today was mainly uh, done. Sorry. I just so let's continue with the last part of your DNA, principle of DNA and isolation of purifications. DNA can be isolated from the nucleated cells. DNA is a giant anion in a solution actually. So sources of DNA includes blood, buccal cells, cultured cells, bacteria, biopsies, forensic samples, that is body fluids, hair follicles, bone and teeth. You can take from any of these parts. So DNA isolation is a routine procedure to collect DNA from subsequent molecular analysis. There are three basic steps in DNA extractions. So first is cell disruption, then isolation of DNA, then precipitating the DNA. So cell disruption, this is the commonly achieved by grinding or sonicating the sample, removing membrane lipids by adding a detergent. Isolation of DNA, removing proteins by adding a protease, then precipitating the DNA. Usually ice cold ethanol or isopropanol is used. Since DNA is insoluble in these alcohols, it will aggregate together, giving a pellet upon centrifugation. This step also removes alcohol soluble salt. So here you can see sample, the cell lysis is done, protein degradation is done, then binding is done, then centrifuge is done again, then washing with the help of wash buffer, then again centrifugation, then elution, then centrifugation, then pure genomic DNA at the end. So basic rules while doing your DNA isolation for the blood. First, lyse, explode the red blood cells with gentle detergents such as Triton X100. Wash cells with hemoglobin, inhibits restriction enzymes and Taq polymerase. Work on ice to slow down enzymatic processes. Wear gloves to protect your samples from you. Autoclave all solutions stored in the fridge. Keep all pellets and supernatant until you have DNA you want. So getting to the DNA. So first you lyse the cells in the presence of sodium chloride, which stabilizes and remains as double helix. Then you do with the EDTA, which chelates magnesium plus and is a cofactor of DNase, which use up a DNA rapidly. Then anionic DNA detergent SDS, which disrupts the lipid layers, helps dissolve membranes, binds positively charged of chromosomal proteins to release the DNA into solutions, which includes a protease to digest proteins, incubated solution at an elevated temperature for 4 to 24 hours. When getting rid of the protein, organic solvent extraction using equal volume phenol is to chloroform, that is 24 is to 1. Protein at the interface of the centrifugation. Then precipitating the DNA at the ice cold 95% ethanol to the DNA, leaving for minus 20 degree overnight. Centrifuge the sample, then again wash your DNA, resuspend in the distilled water, then store at 4 degrees Celsius. And this is the amount of DNA can be quantified with the formula of DNA concentration is microgram per ml, with this OD at 260 optical density, multiply by the 100 dilution factor, multiply by 50 microgram per ml, and divided by 1000. Then you can have the peak absorbance in the ultraviolet range of about 260 nanometer. The purity of DNA is reflected on the OD 260 is to OD 280 ratio must be between 1.6 to 2. If it is less than 1.6, then protein contaminated. If it is more than 2, then chloroform phenol contaminated. Then repurify your sample. So in summary, sample of DNA extraction is done with the help of lysis of cell at elevated temperature, detergent enzyme in the salt buffer, removal of cellular proteins, precipitation of nucleic acid with ethanol, quantitation and purity of measurement of DNA. So for development of this DNA could be done for the development of agriculture development and environmental protection to ensure food security for ever-growing human population. So thank you very much for today's lecture. Yes, yes. So let's check DNA extraction done in the virtual lab. So what we have done here, so we will check the same thing in the virtual lab, how it is done, all what we have learned here with the DNA isolation. We will learn. Uh, so I was fast because I am afraid that networking case could go and go, and I will be not able to teach you everything. So I will make sure that I will be not that fast I was today. I was just thinking that uh, yeah, that everything is done on the time and things are not wasted out again. So sorry if I was fast, but I will try to be in normal pace again. No worries. DNA extraction, biotechnique, virtual lab. Let me have a sip of water. Password दे दो. Yeah, I will send you all the PPTs. Whatever we have done, maybe I can just send you now. Maybe. So let's continue with the virtual lab of DNA extraction. So in the experiment, you need to isolate DNA from a test subjects, uh, and you need to extract human DNA in that case. So scientists isolate DNA for a variety of reasons, some of which includes genetic testing, body identification, analysis of forensic evidence. So let's start with this. We can see in the cell there is a nucleus. In the nucleus there are chromosomes. In the chromosomes we have these bead-like strings, histones. Around histones this DNA uh, are arranged. The skin on the inside of our mouth has thousands of cells every day, and we are here after for them. So let's collect cheek cells, burst open cells to release DNA, then separate DNA from proteins, then isolate concentrated DNA. So in these four steps, we will isolate our DNA. So here the materials equipment you will need to purify DNA from cheek cells. Warm water bath, centrifuge, buccal swab, micropipeters, lysis solution, concentrated salt solution, resuspension buffer, ethanol, isopropyl alcohol. So drag the swab over inside of the test subjects to collect the sample. So I will take them and put them in the mouth and cut. So you have your cheek cells here, and we'll put them in the ependome and put some lysis solution into that. And then put them into the warm water bath. So your histones will be break out because of the detergent and enzyme called proteinase K, and detergent disrupts the cell's membrane and nuclear envelope, causing the cells to burst open and release their DNA. The DNA is still wrapped very tightly around proteins called histones, and protein K cuts down it like this. We have concentrated salt solution, and we put them into the tube. So salt causes proteins and other cellular debris to clump together. Place the tube into the centrifuge. So in order to balance, we put another side one tube also. So, so you have your DNA and liquid on top and protein and debris to be down. We close it and we extract our DNA from the top and put into this ependome. This is isopropyl alcohol. Add some into the tube. So your DNA will coagulate together, and you can see it with the naked eye in the presence of isopropyl alcohol because it does not dissolve in that. That's why we added to that. So we will again centrifuge this so that we can have at the bottom close it. So at the end, at the bottom is your DNA, and rest is just waste. So you can freeze it in the in -4 degrees Celsius for the longer term, as per your needs. This is the last one. So for today, just try to make assignment if possible, uh, and then assignment is possible. Just try to send 2 page assignment that I have shared with you and review articles. Please make a group and start working on it. And I will share you with some free journals where you can publish that also in order to work with it. So thank you very much, everyone. So tomorrow we will meet again. So at the same time, 5 p.m. With so this is the bache, the link that I have shared is the videos link actually. So you can go through the same link that I have shared. That is the link itself. You can watch it through it. Yes, yes, you can take two topics the same person. The topic should be different. That have to take care. Okay, students, thank you very much for today's session. I will see you tomorrow, same time, same place, with more some interesting topics, more virtual labs, and more exciting things for you to keep it better, 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 and something new for you. Okay, it was lovely having this class with you. Sorry for some small hiccups and hitchups things happen today. Maybe make sure that does not happen tomorrow again. So take care. Bye bye. See you soon. You all are awesome. Believe me, you all are awesome. And keep smiling and stay safe. Good night. Bye. हाँ जी हाँ जी